afternoon. My name is Elliot Goldstein. I'm the President and Chief Executive of ProMIS Neurosciences. We're publicly traded on the main board of the Toronto Stock Exchange, the TSX, under the symbol PMN, and on the OTCQB in the United States under the symbol ARFXF. While you're just kind of looking at the obligatory forward-looking statement, let me just say that we're focused entirely on the development, on the identification and development of antibodies that are uniquely selective only for the misfolded forms of toxic proteins that are implicated in the development of neurodegenerative diseases. We launched the company in 2014, really after we saw that what we believed were the stunning first ever positive effect on cognitive decline in Alzheimer's presented by Biogen in their so-called phase 1b study. It was really a phase 2 study in our opinion. Anyway, and that was further confirmed with phase 3. But when we looked at those data, we said something's going on for the first time ever after more than a decade or two of failures targeting amyloid beta, and ultimately we've learned most companies were targeting wrong forms of amyloid beta. We were more than intrigued. We said there's something here, but we should be able to do much better. So we used our proprietary development discovery platform to enhance the antibody so that it's called PMN 310, so it's highly selective only for the misfolded toxic form, the toxic oligomer of amyloid beta. The objective here was to move away from plaque binding because we know plaque is non-toxic now, and we'll discuss that a little bit in a second, but also because products that engage plaque, antibodies that engage plaque, are associated with ARIA-E or edema, which is a dose-limiting side effect, and is really the issue with aducanumab. If they could dose up higher, we'd see more efficacy. Then we used the same proprietary discovery platform based on two patented thermodynamic algorithms. I'll tell you a bit more about how they work in a second, but then to spread that to other misfolded proteins implicated also in various neurodegenerative diseases. The opportunity is huge here because these are the only chronic diseases, at least to my knowledge as an endocrinologist actually, as also a practicing physician, it's the only chronic diseases for which there's no underlying root cause therapy. Nothing, absolutely nothing. You could argue that there's some fairly effective symptomatic treatment for Parkinson's disease, but nevertheless the underlying neurotoxicity and death of neurons continues. The little bubbles here indicate more or less the sort of relative market size. Alzheimer's, 50 million patients diagnosed worldwide. There's another 50 million who have the disease. They just don't know it because they're in the prodromal phase. So basically what you see are a series of neurodegenerative diseases, but actually there's a host of misfolded protein diseases outside of neurodegen. In schizophrenia, for example, DISC1 has been implicated. The amylin, misfolded toxic form of amylin, implicated in certain types of type 2 diabetes. Alpha-synuclein is implicated in a host of Parkinson-like syndromes, not just Parkinson's disease. And of course, we have the originator of all of this, the prion protein, or PRP, which we all have. So this seems to be sort of a general bad thing, if I can say it that way. Proteins gone bad, killing cells, neurons in this case, in their vicinity. Here's a quick snapshot, and I'm only going to show a few things on this, of our portfolio. But as I mentioned, we're targeting now several misfolded proteins, not just the misfolded toxic oligomers of amyloid beta for Alzheimer's, alpha-synuclein I mentioned for the Parkinson's-like syndromes, including Lewy body dementia, TDP-43, which is present in every nucleated cell in the body. Its aggregated form, intracellular and extracellularly, is responsible for prion-like propagation and death of neurons in both ALS and frontotemporal dementia in particular. Tau, of course, we all know its role in various dementias, not just Alzheimer's, but also chronic traumatic encephalopathy. And we have some ongoing discovery research on some of these newer, if you like, and less well-known targets. How does our system work? Well, really the essence of it is we begin with two proprietary thermodynamic algorithms. We have 45 patents issued worldwide and another 40 pending across our entire portfolio. We have a chief physics officer as well as a chief scientific officer, and together they created these algorithms which run on the West Tech supercomputer network here on the West Coast. What's interesting about this is that actually PMN 310, our lead product, targets an epitope, a sequence of amyloid beta called HHQK. Okay, nothing special there. However, the real special feature of our predictive algorithms, it allows us to identify not only the sequence where the protein misfolds, Misfolded, imagine a piece, sweater that gets caught on a nail, a little loop sticks out. These misfolded portions are hydrophobic and they're highly toxic. They're highly wrong, if you like, and very immunogenic. Anyway, our algorithms allow us to identify not only where the misfolding occurs, the sequence, but also the shape of this misfolding. It's like a twisted half circle. To create the antibodies, we synthesize, we have a company synthesized for us in Sunnyvale, California, synthesizes this sort of cyclical peptide, this small series of 4 or 5 amino acids that I mentioned, but in the correct conformation. We inject that into mice. The antibodies, the dozens, if not hundreds of antibody clones that emerge, are highly selective only for the toxic misfolded form. This is a bit of a signature on the misfolded form. It's like a tattoo. If you immunize against just a linear sequence, HHQK, you end up making antibodies that target all forms of amyloid beta, and you have no selectivity, no specificity, like most of the anti-amyloid drugs that were developed early on. Finally, there are several steps to confirm the profile of the product, not only its selective binding profile, but importantly, functionally, does it do what it's supposed to do, stop neurotoxicity and block the propagation. One of our final tests, though, is that we like to see how the antibody performs against the real thing. So in Alzheimer's disease, we use extracts, soluble extracts, brain extracts from patients who've died of AD, and we look at the binding of the various antibodies to see which gives us the most specific, most selective, and highest degree of binding to the real thing. And the misfolded toxic oligomers are contained in the 70 kilodalton and lighter, the smaller fraction of the soluble extract from the brains. So let's talk a little bit about the amyloid hypothesis. Well, the bottom line, it was wrong for 20 years because it was all about plaque, and plaque's not the target. There's no question in our mind and that of many others, over 2,000 publications on misfolded proteins and their toxic roles in various neurodegenerative diseases, that amyloid plays a role in pathology. It may not play the only role, there are other factors, but it certainly plays an important, essential role. So the real question is, can neutralizing amyloid beta, or at least the correct forms of it, be clinically beneficial in symptomatic patients? Or how late or how early in the disease does it make sense? Are other targets more important, et cetera? The aducanumab pivotal data indicate, the emergent engaged trials indicate that it does provide blocking of the toxic soluble form, the toxic misfolded form of amyloid beta, provides symptomatic benefit, and we'll take a look at the data. One problem, though, is when Biogen, you're all familiar with this, right? It's really thrown there are mistakes, if you like, in terms of communication and management decisions in the trial. They've really thrown ice water on the field beginning in March, and then that's translated further on. So, but if you take that into consideration and really look at the so-called V4 analysis, which is really the analysis that included all of the patients who received the highest dose, the highest tolerated dose, 10 mg per kg, you actually see that the results were quite positive, and we'll take a look at those. Just continuing there, what was the big difference between aducanumab and BAN2401 compared to all the other amyloid-directed programs that failed? Well, those two compounds do not bind amyloid monomer, the single-stranded form. Every product that's bound monomer, regardless of what else it does, has failed. They've all been failures. And I'll show you some data on that. It binds plaque. It was designed as, it was developed as a plaque binder, but luckily it also binds to some extent, not as well as we'd like, but to some extent the toxic misfolded oligomers, hence its activity. Both compounds, BAN2401 and Biogen's aducanumab, showed a strong dose response curve, and based on all the clinical and scientific data to date, we believe that a product that, whether it's a small molecule or an antibody or a vaccine for that matter, that avoids plaque binding with strong binding only to ABOs, amyloid beta oligomers, would likely have greater clinical benefit. Without binding plaque, we avoid the ARIA-E, the edema side effect, which allows much higher dosing and greater potency. We believe that our PMN310 potentially is best in class in this respect, and I'll show you some data shortly. Okay, so beginning, as I said, around late 2014, when we saw the first results from Biogen, that's where we evaluated the positive PRIME data set. That was the name of that phase 1b/2 trial, and approached it from the misfolded protein standpoint. We then also used our proprietary discovery platform to develop other to develop PMN310 with selective binding for ABOs, and also then expanded it to other misfolded proteins, as I mentioned. Interesting, though, the 3 boxes here is really the cold ice water that was thrown on the whole field by Biogen's, I'll have to say, repeated issues, if you like, repeated miscommunications, including accidentally disclosing an increase of 500 patients without a reasonable explanation, if you like, which just created uncertainty and questions. ESI really flubbed, if I can use that word, that non-technical word, created a lot of confusion over ApoE4, patients who were ApoE4 carriers and their results. And then finally, the erroneous futility trial, because the futility was based on an incomplete data set because of the modifications to the protocol. But nevertheless, despite that negative sentiment, which is sort of correcting now, it's not completely over, we believe that 2020 will mark really a big change in the field. Oh, yeah. So what are their clinical data and their preclinical scientific data, I think that absolutely nail the answer to the question, is amyloid toxic to neurons in Alzheimer's disease, and does it play a significant role? So let's look at the clinical data. Quickly from the PRIME study, as you can see, there's a dose response, there's sort of a cartoon, if you like, but these data were taken from their publication. A placebo down to 10 milligrams per kilogram. That's the highest tolerated dose, with about 30% to 50% of patients developing ARIA-E or edema, but with it's statistically significant and clinically significant, but moderate treatment effect after 52 weeks. In the V4 version of the pivotal program, the emerging engaged trial, so these are the data that include the patients on high dose, we see very similar results with a strong statistical significance, a treatment effect in the high 20s to 30% on CDR sum of boxes, and interestingly enough, and actually more encouraging for the clinician, is that in the eMERGE trial, there's a 40% difference with respect to placebo in terms of activities of daily living, and that's what really counts in early Alzheimer's disease, the ability to conduct yourself during the day. So we believe there's strong scientific data, or clinical data here, showing that targeting amyloid is the right thing. So the 3 basic forms of amyloid, as many of you know: monomer, single strips. Oligomer just means more than one, so these are sticky proteins. They chemically clump together and form oligomers of 2, 4, 10, 12, 20, sometimes up to 50 or 100 mer, if you like, all stuck together. Eventually, these twist into fibrils or fibrils, depending on where you learned your English, and that constitutes plaque. Only one of these, though, is toxic. So let's just look at the history of all the failures, Over 20 phase 2 and 3 trials targeting monomer, whether they were BACE inhibitors, solanezumab, or others. Any product that had targeting of monomer that showed binding to monomer failed. That's just a simple factual conclusion. You can interpret it the way you want. None of those trials were positive. In addition, there were 10 phase 2 and 3 clinical trials that non-selectively targeted all forms of amyloid beta, like bapineuzumab. They all failed as well, mainly because they're wasting so much ammunition off target, they have no chance in hell of working because the oligomers are present in very low concentration. And the only 2 products with 3 positive trials are BAN2401 phase 2 and the 2 trial programs from Biogen, their phase 1b prime, and then their pivotal program. What's the scientific data? So there's a clinical data set. That's what we learned from looking at all the results of clinical trials. What about what we've done in the lab? This experiment on the left is these are human hippocampal neurons grown in culture. We can do that regularly now. We've done this experiment. Many others have as well. If you put monomer into this system, you see an absolutely normal neuron at 24 hours. If you put fibrillar forms of amyloid beta in, i.e. plaque, the same thing. You have to use synthetic toxic oligomers here because the natural ones are just unstable. If you put in synthetic toxic oligomers, in 6 hours the neuron is dying. At 24 hours, there's just the skeletal remain. Similarly, in the dual object recognition test, which is well known to most people in the CNS area, there's your normal response. I won't go into the details of how this works, but there's a normal response. It's between 0.35 and 0.5. Monomers put in here in the intracerebral space, the intraventricular space in the brain of a mouse. No change in the response. Likewise, putting fibrils into this space, no response. Oligomers, the behavioral response and the biochemical response in the dissected hippocampus completely destroyed. This is really nasty foreign stuff. That's really the conclusion here. Okay, so if we answered the question, is amyloid beta a valid target? Yes. The proof is from these clinical trials, but which form is the right target? The clinical trials targeting monomer all failed, as I said. Plaque's not the pathogenic species. It's the misfolded toxic forms. The misfolded region is hydrophobic. It's normally buried inside the molecule. When it sticks out, like when you catch your sweater on a nail, that little loop, that's a hydrophobic loop, which is highly toxic to any cell in its environs. The stickiness, if you like, allows them to propagate in a prion-like fashion. So targeting toxic ABOs does matter. Aducanumab showed dose response with the 10 mg per kg as an effective dose. The dose-limiting adverse event or side effect prohibits higher dosing. What a shame. If they could go up higher, we'd probably see greater potency and efficacy. That's why we believe that PMN 310, with its selective binding of toxic ABOs, should allow higher dosing and greater therapeutic potential. So I'll quickly run through some data slides to hopefully prove the point. I show this a dot plot. It's not the most powerful scientific experiment, but it's visually sort of interesting. So what you do on a special membrane is you put down a microscopic dot of monomers, in this case monomers of amyloid beta, fibrils, which are the constituent of plaque, and these toxic oligomers. Okay, they're just sitting there. Then you place, you just place a microspot of your antibody on top, and it's not quantitative, it's just qualitative. If it lights up dark like this, it means there's strong, much greater than background binding. So bapineuzumab, which binds all forms, total failure. Solanezumab, you see here, only binds monomer. This is really background. So again, total failure. We've compared our PMN 310 to just about every antibody or amyloid targeting antibody that's been in development preclinically and clinically. They're available from Creative Biolabs in China, by the way, if you're interested in getting them. Here you can see aducanumab, and BAN2401 shows the same profile. It's slightly better here, actually. You can see binding of fibrils and plaque. That's what it was intended to do. But also a dark circle here on the toxic oligomers, hence its efficacy. And PMN 310, we just see background noise here. It only binds toxic oligomers. Okay, what about plaque binding? This immunohistochemistry, the staining for binding to plaque, you can see here. These cuts are from the same brain, from the same Alzheimer's brain of a patient who, in the same region of the brain. This is the hippocampal part of, no, it's frontal cortex, I think. Anyway, you can see here the parenchymal plaque binding, these dark brown squares. And importantly, look at this perivascular binding. That's what's believed to be at least a component of the edema. As you engage or bust up the plaque, you open up small channels or pores in the vessels, and fluid leaks out. You've got edema. Same slice, like completely clean, no binding to fibrillar structures or plaque anywhere with PMN 310. Final test, as I mentioned, we really like to see how our products perform against the real thing from soluble extract of brains. This is, as you can see, soluble extracts from 11 different brains. And as you can see, the binding is measured by SPR or Biocore, if you like. We use a different machine, but it's the same process. Using SPR, you can see here on the left panel, much greater binding of the humanized PMN 310 versus aducanumab, bapineuzumab, and this is just a control. And remember, all of the product of PMN 310 that crosses the blood-brain barrier can only bind toxic oligomers. So it's not like any of the ammunition, if you like, is wasted on off targets such as plaque or monomer. Just a note that the world's largest selling products are best in class, not first in class today, whether it's Lipitor, Humira, the largest selling antibody, $16 billion annually. It's just gone off patent. And Sovaldi, of course, which sold $25 billion in its first 12 months on the market. And we believe we are developing a best-in-class product here, having engineered around some of the liabilities with aducanumab and BAN2401. Right now, in terms of the status of the program, we finished the cell line development. That's gone beautifully. We've done that with Selexis in Geneva. So we need to raise capital to actually finish the scale-up manufacturing preclinical talks. And here's sort of just a very rough outline of some of the things we want to look at. We're very interested in Alzheimer's disease in Down syndrome because, as you know, because they have 3 copies of chromosome 21, they have a very high amyloid overload because the gene that codes for APP sits on chromosome 21. They have 3 copies. It's a completely different pathology than the intellectual decline seen in that disease. Okay, 2 final slides just to conclude. Number of catalytic events we believe will be positive for the field in general and hopefully for us. External events, Biogen's BLA filing, we anticipated sometime this quarter probably, according to the news we've heard. That'll be followed up with advisory committee and hopefully approval late this year, early next year, along with BAN2401. For PMN 310, we would anticipate having potential to actually compare in phase 1 with biomarker study, for example, using fluid-based NFL, neurofilament light, as a good marker of neurodegeneration. We could do comparative trial. As I mentioned, we're very interested in the Down syndrome, one, because of the unmet need in that population, but importantly because of just the heavy amyloid charge. They develop Alzheimer's symptomatology as early as the teens, and by the age of 55 or 60, 75 to almost 100% of Down syndrome subjects have Alzheimer's disease. In conclusion then, we launched the company based really on what we saw with the stunning data from Biogen. Our lead program, PMN 310, based on the use of our unique proprietary platform, allowed us to make improvements over Biogen. We believe Biogen's aducanumab is approvable but improvable, and that's what we've done. We'll capitalize on fluid-based biomarkers for rapid go, no-go proof of concept in phase 1 in patients. That can be done. And our unique capability to develop these highly selective antibodies is also being applied to other neurodegenerative diseases like ALS, Parkinson's disease, and we're in active partnering discussions with multiple large pharma companies in that respect. And I think my time is up, and I want to just thank you very much for your kind attention.